Okay, thank you very much, everyone. Thank you, thank you all the organizers. It's a pleasure to be here. I love this meeting. I should be a bit careful saying that because it's recorded, and you can go and check all the other meetings I go to and find out I always say that it's the best meeting. I really, last year enjoyed this meeting tremendously, and I found it to be a really special meeting because of its emphasis on methods development. Since I'm the first speaker, just two things I'll say. The first is this is a tutorial, and I tried really hard last year, and I'll do the same this year, to really actually make this a tutorial. So I'm going to go pretty slow and through material that is partly very, at the level of teaching, there's concepts rather than showcasing my research, although I'll talk a little bit about research towards the end, but we'll try to go slow, and if you have any questions, please ask me. The second is that I was told, and you'll find out, that the speakers introduce themselves. Here's my introduction. My name is Lior, and like most tenured professors in this field, I'm a professor of Twitter, and so most of what I do all day is surf Twitter, and that's how I'll start. I actually love Twitter because I learn a lot of math and statistics and biology there. And this week I came upon an interesting little tweet with a little problem that is in some way actually kind of pertinent to this talk, so I thought I'd start it as a warm-up. So this is from Republic of Math, which has 75,000 Twitter followers, and a great account to follow for lots of fun, sort of what you might call recreational mathematics. And the question here was that, you have a circle, and you're going to place 6 circles inside it, and you get to choose their size, but they touch each other end to end. And the question is, what size should they be so that the total area of the internal circles is minimized? Or is there even a minimum to attain, and what would it be? And I thought this tweet was interesting because it's not the 90s anymore. In data science here, even the mathematicians are on board, and first thing you do is actually simulate it, and you get 0.5236, so you guess something like a half. So I thought I'd start with that question. Anybody know the answer off the top of their head? Any guesses what the answer is? No guesses. It is early in the morning, but I'm very patient. So I could surf some Twitter while I wait. Okay, sorry?
Is it equal what?
Equal?
Equal radius.
Equal radius, yes, that's the right answer. Excellent, somebody said equal radius. Why is the answer equal radius? It's good intuition. Any proofs?
Minimizing the sum of the squared radii. Minimizing the sum of the squared radii.
Yes.
And the constraint is on the sum of the radii.
Correct, so you're minimizing the sum of the squares of the radius subject to the sum equals constant, yes.
So that works at the end point, where the radii are equal.
Okay. So that's right. So it's sort of true by tautology because it sort of has to be true. So it's interesting. I won't shame anyone, but if you go and look at the answers, these are all math people. They figured it out. This is the wrong answer, 0.5236. I'm sorry, the wrong answer is a half. The right answer is 0.5236. The way you do this little calculus problem is use this horrible thing called Lagrange multipliers, or you do there's something called the root mean square arithmetic mean inequality. But because I do statistics and I was preparing this talk for today, I'm going to show you how I think you should think about it, and what's sort of the value in like understanding PCA and CCA, which is going to be my topic for today. So the first thing you need to do is you need to replace the D's by P's. That's very important. So, and the reason is that instead of diameters adding up to 2, you want that P's always add up to 1, as you know. And maybe we'll also turn it to n. We might as well, instead of doing 6. And so you see, when you are doing PCA all day and CCA and statistics, and you know probability sum to 1, and you're minimizing squares, then the first thing you think about is variance. And so actually, you realize that minimizing the sum of the squares of these PIs is equivalent to minimizing the variance, which I'll just write like this. Because if the PI sum to 1, then the mean is 1 over n. And why is this true? Well, I'll let you think about it for a second, but if you square this, it's a constant, and then you get the sum of the p's equal to 1, that sort of cancels out. So really what you're doing is you're trying to minimize this quantity, and this quantity is obviously minimized when pi is equal to 1 over n for each I. Because then these terms are 0. The smallest of variance can be 0. And I mention this because this is basically the magic of everything I'll talk about today. It's right there in this math problem. Is that when you minimize a sum of squares, I'm sorry, I forgot to put the square here, subject to a constraint like this, and so it actually turns out to be easy. You don't need all the complicated machinery of calculus and all these things. And this magic is what allows PCA to work, and actually also basically what allows CCA to work, which is my topic of my talk. So the take-home message here is minimizing sum of squares is subject to a constraint like this, is easy if you think like a computational biologist. All right, so I'm going to move on and actually get to the topic of the talk. I'll just preface it by saying that this is a subject I've thought about for a couple of years now for a number of different applications, and a lot of my thinking has been shaped by Shannon McCurdy, who's just wrapping up her postdoc with me, and my former graduate students, Breland Brown, currently working at Verily, and Nicholas Bray, who's a postdoc just starting up with the Aviv Regev group. So we also have been thinking about canonical correlation analysis for a while, and sort of my viewpoint on it, as I said, shaped by them. So let me get to the topic. So CCA is an abbreviation for canonical correlation analysis. And this is a kind of standard statistical technique that really all of you should know, because it's sort of as straightforward and basic and classical as any other kind of statistical technique, such as principal component analysis. It was sort of introduced by Harold Hoteling, I think in the mid-30s. This is actually a quote from his paper. His sort of mindset in introducing this was an application of shooting, even back then, what do you know? You have two marksmen side by side. Sort of the kind of, the application here is that, well, there are sort of like independent random factors affecting their shots. They're very nervous when they shoot, so they shake a little bit. But then there's effects such as wind, which is affecting both of their shots. And so when you look at the data, you kind of want to take into account the sort of shared effects while allowing for modeling of independent effects. And he noted that people ordinarily look at sort of one component, but he wanted to look at a multivariate version of this. So he introduced canonical correlation analysis. Principal component analysis, which I think more of you probably have heard of, was also introduced by Hoteling, I think just 2 or 3 years before. It actually was done by Carl Pearson early in the 20th century, but they Hoteling didn't know this and did it independently. And so the point I'm trying to make is that this is a classical technique. However, it's not so widely used in our domain of genomics. It has been used, and a lot of practitioners know it and use it, but it's sort of a kind of a dark sibling, if you like, to PCA. And if I had to give a purpose to this tutorial is to introduce more of you to this technique and have you believe that it's a useful tool that should be in your toolkit. And like component analysis, it is useful for exploratory data analysis. It's sort of a useful tool to look at your data. It has a statistical interpretation in a similar way to principal component analysis. And the method by which it works turns out to be the same magic of singular value decomposition. So that, and it's in a way a real analog of principal component analysis, and the similarity is not just superficial. It's deeper than that. I mean, these techniques are really very much closely related because they involve essentially related models. And so it has been used sporadically in genomics. It's actually a technique more widely used in other areas such as social science, not so much in genomics, but statisticians working in genomics definitely know about CCA. And there are many examples I could put here, but two papers I thought I'd highlight are work by Daniela Witten. She has done a number of really nice both theory papers, but also has really nice software and R implementing a lot of variety of different flavors of canonical correlation analysis. Charlotte Sonneson is also a statistician, wrote a very nice paper examining also a lot of flavors of canonical correlation analysis around the same time as Daniela, with a more applied bent, looking specifically at questions in genomics and how this technique is useful for multimodal data analysis. That is, data analysis where you have data sets of different types for the same system, and you're trying to do an integrative analysis. I'll point out that that kind of analysis is becoming much more commonplace in high dimension as we have more and more sequencing-based technologies producing data sets for the same system or even for the same cell of many different modalities. Two applications of that, so that you know that I'm not just making this up, but it's sort of for real, is there's a paper from earlier this year demonstrating how to use canonical correlation analysis for integrating single-cell RNA-seq data sets. I think single-cell RNA-seq will be one of the topics in one of the journal clubs at this meeting and probably some of the talks, so you might find that interesting. On the level of omics data integration, there's a nice paper by Jandubi and Corby and Strimmer, which likewise demonstrates how CCA can be viewed as a really useful tool for integrative analysis. And they do a very nice theoretical development of a sort of probabilistic framework for CCA, which is something people have done previously, but is not as worked out as the PCA framework. So that's a little bit of the sort of background. I think that, if I had to make one prediction in the lecture, my tutorial today is that I think you'll see many, many more examples of this because CCA is such a quick and easy technique to use like PCA. It's not perfect. The model, as you'll see, makes various assumptions about the data, but to first order, it's a useful tool. So in order to explain what canonical correlation really is, I'm going to start with principal component analysis, its sort of sibling. And I don't have that much time today, so I probably won't get into all the details of CCA, but I'm here all week and actually next week. So part of this tutorial will be just an introduction, and I'll be happy to meet with you all and talk more if you have questions later. So I'm just going to go briefly through these three aspects of CCA, namely the tool, its view as an inference algorithm, and the actual method by which it's done, starting with PCA so that you can get a grip on sort of what exactly I mean by these things. And I'll start by saying that in PCA, the input is a matrix, and the output is a picture like this with some dots. So that's why it's useful sort of to explore data, because you can put any matrix into it and you get a little picture out. And the picture is nice to look at. So it's nicer to look at than a table of numbers. So let me talk about how it is really a tool. And to do that, I want to give an example from genetics where people use PCA for genotype matrices. And by that, I mean a matrix where you have the rows label individuals and the columns label their genotype at a site in their genome. And these matrices have zeros, ones, and twos because there's an encoding, which is that most polymorphic sites in the genome are biallelic, meaning that you have either, let's say an A or a G. You don't have any of the 4 possible nucleotides appearing, and you have a pair of chromosomes, and so you'll be either AA or GG or AG or GA. And if you're homozygous, let's say for A, you'll get a 0, and if you're homozygous for G, you'll get a 2, and if you're heterozygous, meaning you're one or the other, you'll get a 1. And these days, people can phase genomes more easily, so they know exactly what each chromosome is. But this kind of representation is useful, especially for unphased data where you just measure independently site by site. And so, if it's A from mom and G from dad, you're not going to distinguish those. You just put a 1. This is a standard encoding. And so people started collecting these matrices a while ago. It became more high throughput when microarrays were introduced. And already about 10 years ago, there were data sets of hundreds, starting to be thousands of individuals where you had these matrices. It's not very pretty to look at a matrix like this. What you can do is put it into this thing, PCA, and get a picture with dots. And there's a very nice paper that's very famous. One of the organizers you saw earlier for CGSI is John November, and this is his famous, I would say, paper, where when he performed PCA on this matrix, he got this picture. This is just a 2D picture, x-axis, y-axis, labeled by these things I'll explain in a second. And each dot here is a person whose vector of zeros, ones, and twos now became a point in the plane. And you should think about this. People use this word projection. You projected the data, but it actually has meaning here because the data, you could think about it like points in a high dimensional space where the dimension is the number of columns here. So it's a point in, hundreds of thousands or millions of dimensions. And literally, PCA is performing a very special projection, as in a linear map, onto the plane. He colored each dot here by where the person was actually from. And if you kind of cross your eyes, you can sort of see the map of Europe, which is pretty interesting. And this turned out to be useful, not just because it obviously is informative from a population genomics point of view to learn about ancestry, but it also turns out to be useful as a way to correct for this kind of structure in data when you do statistical testing for things like genome-wide association analysis. And I would characterize this as sort of exploratory data analysis because literally you took the matrix, you put it into the tool. There's no particular sense in which you're worrying too much about the model or anything else. So, there's no like coalescent theory here or something. It's just a tool for looking at the data. And to give you an understanding of sort of why it works, so what is it doing really, it's sort of nice to go to 2 dimensions. So, instead of being in 100,000 dimensions and going to 2D, I like to go from 2D to 1D. Now, you have to be kind of careful because sometimes intuition in 2 dimensions doesn't work in 5 dimensions, let alone 100,000. But for this purpose, it's sort of legitimate. You'll have to take my word for it. So what's happening here is you have these points. These are the individuals. Now they have just 2 SNPs. And so that's why the dimension is 2. And you're trying to find a line to project them onto that achieves this, that minimizes the sum of the squares of the distances from the points straight to the line. So if I was to choose this line, I would go like that. And you see, it looks a little bit like that, okay? Yeah. So I'm minimizing the sum of the squares. And this line isn't just any line. It has to go through this big green fat dot, which is right at the center of the points. Like its coordinate is the average of the x coordinates and the average of the y coordinates. People actually don't usually realize this or say it, but this point is called zero-dimensional PCA. The mean, the average, is zero-dimensional PCA. It's right there, over here, 1/n. And then this line is the one-dimensional PCA. So you're choosing a line that's tethered or going through this green dot to minimize the sum of the squares. So one way to think about this PCA, you're trying to sort of find a line, or a plane, in the case of the the genotypes here, that's close to all your points, all right? But the reason that this picture kind of works is because of an analogous interpretation, which is that instead of thinking about the line that minimizes the sum of the squares of the points to this line, you can think about maximizing the distance between your point and the fat green dot. And that's the variance of your data. And those are equivalent because each point is a fixed distance from the fat green dot, which is the mean. And because this squared plus this squared equals that squared, yeah, Pythagoras' theorem, then minimizing this is the same as maximizing that. And so another way to think about John's picture is that he's not just trying to find the plane that's close to all the genotypes. He's trying to find the plane that maximally represents all the variation among people. And that's actually a really good way to think about why this is showing the variation among people, right? Because it's designed to do that. All right, so that's sort of the two-dimensional intuition. And one of the things I want to say is that as a tool, it's been very, very useful for precisely this reason. Often you have this complicated data set, and you want to see what inside it is really variable. You do this PCA thing. But it is also true that this PCA thing is not just some magical procedure that does this minimization. It has a statistical interpretation. I think this is very important, and I'll show you why in a second. And the interpretation is actually very simple. So instead of just thinking about data on which I find this projection onto some line I choose, you have to sort of shift your mindset and think about making the data. We're going to create data by taking an actual line, which is the true line, and we're going to randomly choose points along it, and I've selected just one right here to show you how it's made. So we're going to randomly choose points here. They're going to be chosen from a Gaussian distribution centered at the origin, so that we're unlikely to choose points far away. We're more likely to choose points close to the middle. And then once we choose a blue point, we're going to perturb it by choosing a pair of random variables from two Gaussians with the same variance in this direction and that direction. And that's why there's a sort of circular fuzz here, because we're picking a random direction. If you pick a random number this way and a random number this way with the same variance, that's equivalent to picking a random direction along the circle and then a distance from a Gaussian distribution and moving over there. And you do this over and over and over, and that's how you make your data. So this is called probabilistic PCA. It's not exactly the same as PCA. It's called probabilistic because there's a probability model, a stochastic model, that is making the data, and the way you think about doing PCA is you're trying to find this line. You don't get to see this line, you only got these red dots. And now you need to find the line. So that's called probabilistic PCA. And let me show you how you actually go about doing it. Well, what you do is you got to find this line, you're given these dots. You're a statistician, they were made by this model. So you pick some line, you put, you have this green tether in the middle, and you smush the points under that line. You project them straight down orthogonally onto that line. And now what you do is you say, okay, that's kind of my best guess for the blue dots that made the red dots. These are the red dots. Those are what I see. But they actually came from blue dots, and those are my best guesses right now. And once I have those blue guesses, now I rotate my line so that they're as close as possible to the original data. But like the blue dots are like tethered after the previous step. So these lines are not flat anymore, like they're not orthogonal. They're, you can imagine like you pin down the blue dots and you're trying to rotate things so that the sum of squares is minimized. And it turns out that's for the same kind of magic as here. You can kind of do that pretty easily. And so you can iterate this algorithm, and you can actually get a guess for this line. And that's called the EM algorithm. So if you've heard about the EM algorithm, that's actually what this is doing. And it's not exactly PCA, but it's sort of in the limit it is PCA. So in the limit that the variance here, by which you do this perturbation, is 0, they're equivalent algorithms. So they're not exactly the same, but they're closely related, and this sort of provides some kind of interpretation to what PCA is actually doing. So why is it important to know this sort of model-based view, and why did I spend my precious time telling you about it? Well, so if you go and think about John's use of PCA as a tool, you know, he applied it to zeros, ones, and twos, and you can just see right away that if you think about this model, you have a plane where the populations are really from, the individuals, and you perturb them with the Gaussian, you're not going to get zeros, ones, and twos. You're going to get some real numbers. And so, you know, it's not quite the right model for this type of data. Now, it works because there's some model misspecification, and it's okay. You know, like you don't have to always do inference with respect to the exact correct model. And it works because there's this kind of qualitative interpretation to it in terms of maximizing the variance in the projection. But you can do better, and there's a nice paper by Yang and co-authors. I think some of the co-authors are in this room, Eleazar and Eskin and Eran Halperin, so you can ask them about it. And if you actually do a sort of version of PCA where you actually model the data as a discrete data, then you get better results. So I won't rehash their paper, but that's why you need to care about the model, because for a specific data instance you're looking at, you might want to tweak the PCA or the CCA, which I'll get to in a second. Okay, the last bit of PCA math I'll do, so that I'll end on time today, is the way you actually do this is you write a command in R. And I actually could have put it up, but I decided not to, because that you can Google. But what you can't actually Google, because Google's not very good at it, is stuff like this. Because everybody uses different symbols, and with the LaTeX, it's hard to Google math, actually. That's just something you find out if you try. So I thought I'd show you the math, because it turns out that if you type PCR comp, there's a function in R, or in Python, all these things, you run a procedure in linear algebra called singular value decomposition. And singular value decomposition is a decomposition of your data matrix into something called a unitary matrix, a matrix of what's called singular values, and another unitary matrix. I'm not going to teach you today singular value decomposition, but it's very easy to see that this matrix here of singular vectors gives you the eigenvectors for what's called the covariance matrix. And this relationship is what actually makes SVD tick work for PCA. And it's like a one-line derivation. I said this word unitary. Unitary means that this cancels. So, if you take X transpose X and, transpose, you just reverse this and put T's everywhere. So it's like, T becomes V, UT, you get that. And then you have to go through the mental exercise, and that could be your homework for today, of, why are the eigenvectors of the covariance matrix, why are they really what I want to do when I'm minimizing the sum of square distances? That's not hard to see. And so that's the one missing part. The other thing is that there's a couple of things going on here. You're looking for this line, but you also want the actual projection. When you're John November and you want to make that picture, you want to draw the dots as they appear on the plane, and that's very easy too. This is the projection, the projected points. They're given by just taking the first couple of columns of these matrices. And so basically, you do this magic, and you have everything you need. The plane, or the linear space in the higher dimension, you have the actual projection, you have everything. And SVD has been highly optimized by mathematicians, and so that's why you can go and type PCA in Python or R in any package you want, and you get the answer fairly quickly. That's why it's a useful tool. All right, so let me get into CCA before I run out of complete time. Before I do that, I just want to say that the figures I just showed, and if you want to play with PCA, I have a blog post about it and also code that implements it that makes the exact figures I just showed you. So, and I think these talks go on YouTube, so you can look at it there afterwards. But if you, I really suggest that you actually go and play with it. So let me get to canonical correlation analysis. Now, if I had time, and I don't, so I will not do it, but I would go through carefully these three aspects of CCA, just like I did PCA, with all the math. And I don't have time to do it in the 45 minutes today, but I plan to, I'm going to like finish it up and post it later so that it's sort of complete as a talk. But for today, I won't do that, and I'll sort of focus on the first to give you some intuition. So the first thing is, what is the input into CCA? Well, it's like PCA, where you put in a matrix, but now you put in two matrices, or three, or four. You put in many different matrices. Actually, this figure is from a recent paper from the Satija group at the New York Genome Center by Andrew Butler. So this is a figure from the paper. You put in two matrices, and you get a picture like this. So it's kind of the same kind of thing, except now the picture shows you both of the matrices. So you have the red points and the blue points from this one. And what the procedure is trying to do here is, unlike PCA, which takes one matrix and sort of shows you the variability inside that data in 2D, here you've sort of tried to glue together these data sets in the best possible way. And I'll make that a little bit more rigorous. What you're trying to do is find two different projections, so one for this data and one for this data, onto the same space, so that the correlation between the points is maximized in this direction, and then once you've done that, the correlation is maximized in this direction. And then you can keep doing that successively. So that's sort of the idea here. Now, a key point about the input, and I want to say that, is that in order for CCA to work, you can't just put any two matrices in. You can't just have points here and there. The points have to have corresponding labels. So in this figure, it says genes here and genes there. This is from single-cell RNA-seq. Each dot here, I'll show you in a second, represents a gene, but each column here represents a cell. Now, in the red data, it's one experiment. All the cells are different than this experiment. But the genes are the same that you made your measurements on. So in this picture here, each point is a dot, and it started off in high dimension. The dimension is the number of cells. And so, this gene has a measurement in its cells here, and in the other experiment, so I'll go here, I'm not hiding from you guys. In the other experiment, this gene has a whole bunch of measurements in some other cells. But the genes correspond to each other. And so here you're trying to put the genes as points on the same picture. Okay? So CCA is useful for multimodal analysis in this sense. When you have corresponding data, you can do this kind of thing. All right. So I just said that in words, but I wrote it out here in text. You're trying to find now a pair of projections that maximize the correlation successively. So first, you look for a projection that maximizes the correlation along this axis, and then a projection that maximizes the correlation. Now, why can I talk about correlation? Well, because the points correspond to each other. So when I project this point and this point, I now have 2 points for which I have a single number, so I can actually make a scatter plot. I'm going to do that here real quick. So you should think about it like this. This is my first gene, and this is my first measurement and my second measurement, right? And then I have here my second gene, and my third gene, and my fourth gene, and so on. And I'm trying to get numbers for them. These x and y values are from the projection so that they're highly correlated. All right, that's what I'm trying to do here. I'm basically trying to glue them together to each other. Sounds kind of complicated, actually. Because first you do it on, your correlation doesn't make sense really naively in 2 dimensions. You can make correlations when you have scatter plots. So you do exactly that. You first project onto 1D, and then once you've done that, then you look for an orthogonal space to the first one you found, which maximizes the correlation in an orthogonal space. And you keep doing that successively. So that's what CCA did. So here's a very simple example I did. So I took a random matrix that I made, like it's a 100 by 100 matrix. I'll show you in a second where I got it. And I wanted to show you that CCA works. So I first put the easiest possible case. I took my first matrix and I just added 10 to each number. So I'm just showing here the values in one matrix versus the other matrix. They're identical, they're just shifted by 10. Because I literally took my first data set and I just added 10 to each value. So if I do CCA, I should be able to figure that out. Because I should be able to find the projection. Projection is trivial. So that the correlation's 1. Now I should basically be able to recover exactly this line. Well, and I can. So I ran CCA, and I plotted my red matrix and my blue matrix. I could have put them on top of each other, but I didn't do that here because, well, I guess I would have to color the points purple. But then, you wouldn't. OK, you see my problem. So I put them separately, and I think I should finish in a minute. OK, hopefully. Yeah, I'll finish. 10 minutes, good. Yeah, I'll finish them. So yeah, you can look eye by eye and you can check, but the numbers are identical. So that's kind of nice, right? Basically CCA realized that the only, that's the wind. The plus 10 on each element, that was the wind here, okay? And so the wind is, each guy got exact, the wind here was perfectly shifted, everything the same. And because I shifted every element, there's no independent noise of the shooters. And so it was trivial for CCA to just glue them straight together. All right, so here's another example where I just took 2 unrelated matrices, random numbers. And now there's no correlation between them. So, there's this shooter shaking like crazy, this shooter shaking like crazy, there's no wind. So I don't learn anything by trying to find projections that maximize the correlation. Every point here landed somewhere. I could have labeled these points, and you'd see that there's no rhyme or reason to it. So it's just a mush. So when the points get clustered together, you've sort of been able to mod out the wind. So where I got all of this, and I call this exploratory methods analysis, is there's a very nice blog post about PCA. It's by Valentine Svensson, who's a postdoc. He's here at this meeting. He goes through the exercise of building a lot of matrices with different structure and looking at what does PCA do to them. He's doing this in the context of single-cell RNA-seq. So I've done a very trivial case of this here for CCA, but it's a good exercise to go and play around and add correlation structure here and there. He has a really nice Python notebook and R Markdown to do that, so it's very easy to do that. And that's where I got these matrices to play around with. So in my last 5 minutes, and then I'll finish, I wanted to show you another, an actual application that comes out of my own research. It's a preprint we just put online, I think about 2 weeks ago or a week ago. And it's an application to expression data. And it's a kind of I think, a nice illustration of one of the things you can do with CCA. And so we were interested in the following question. So this very pretty picture, people have tried to reproduce it, but not using genotypes, but using expression measurements, where they measure RNA. So now instead of a 0, 1, or 2 for everybody, for a SNP, for every gene, say 20,000 of them, you have some number. How expressed is it? And you do this for people from different parts of the world. And this was done already a number of years ago. This is from a paper from the Dermatsakis group. And you got a picture like this. And you really need to, your eyes, it's not your eyes. It just doesn't work, okay? So you can't, there's no real structure here. they comment, there's a little bit of light green over here. By the way, these are labels for populations. YRI are Africans, these are Mexicans, Japanese, and so on. So there's some little bit of structure, but not really. And we were interested in the question, is there a projection that does reveal population structure? Now, there's a lot of reasons that they hypothesize and examine why you don't find population structure here. Well, basically it's because you're trying to project the data to maximize the variance in the projection, and you can have variance due to all sorts of reasons. No, one of them is, population structure, but in genotype data, that's all there is. Genotype measurements are very accurate, and in the expression data, it's a mess. There's like a lot of noise in the measurements. Genes have all sorts of issues. So, there's lots of reasons why you might not find PCA revealing it, but it's still there. So I'll go through this quickly. There's a very nice data set from the Lapalainen group, also at the New York Genome Center. She collected, I think, data from 462 individuals from around the world, where you have their genotypes, actually even more than that, you have their haplotypes, but you have the genetic data, but also she did measurements of expression from lymphoid blastoid cell lines from these individuals. We did an analysis of it. We looked at only 370 individuals. We left out this group because it turns out that cell lines go awry when they're old, and this one was old and had like some strange structure in it, so we omitted it. Okay, so first of all, we can kind of recapitulate the results from Barbara Stranger. So if you look at this plot, if you just do PCA, these are the Africans. Again, green is a little bit, I'm wearing green, a little bit of an outlier, but it's not really structured. One other nice thing is you can take the genotypes and do the same thing, and the November paper is correct and accurate, it works. You find beautiful population structure. So it's not an expression data in the PCA, it is here, so our idea was let's do CCA. It's not such a great idea, but it's an interesting idea because by doing that, you might be able to find a projection that's sort of gluing together the expression data into this population structure to reveal it. So we did that. And what you find when you do that is, a picture like this. So this is the actual CCA picture. In order to do this, and this is a key point, and not overfit, I'll explain in a second, we first had to do PCA on the data sets, and then CCA. So there's a kind of two-step process here. Because it turns out you have to be a little bit careful. If you CCA two data sets, like I just did here, maybe you can always find structure like this. Maybe it's just the result of geometry in high dimensions and has nothing to do with population structure. So we were very careful and checked that. To explain why, and I didn't bring my D&D dice, but maybe that's a game we can play this week. So if you have a tetrahedron and you project it onto the plane, you'll see that you can always find an orientation so that any dot goes in the middle. All right, it's not hard to see you can do that. So if I show you a picture of a tetrahedron and say, look, I could put purple in the middle, that's not a big deal. Like I could put any dot there. You have to be careful. And actually, this is kind of a fun exercise. If you CCA these individuals against their populations in high dimension, there are so many projections to choose from. You remember, you get to choose two projections, any ones you want, to maximize the correlation, that you can actually get this picture. Each one of these dots is a lot of dots. They're all collapsed onto each other. That's the case over here, where the variance is 0, because you have the same point. And so I could actually cheat, right? I could show, look, perfect population structure, but it's for no other reason than I just have a lot of projections to choose from. But if you PCA first, then you limit that, and you can make this a result that has some sort of statistical meaning. More on my last slide. I'll just say a few things. So there's a machine learning technique called linear discriminant analysis. It's another nice exercise to work out why that's a special case of CCA, and that's in our preprint. I haven't talked about the inference parts or the mechanics, but it turns out that CCA instead of looking for the eigenvectors of XX transpose, you now have a matrix X and Y. You take XY transpose. It's called the cross-covariance matrix. And to do that, you just take the singular values decomposition of X and the singular value decomposition of Y. And so it's just as easy to do this as PCA. And it is not an accident, actually, that that's the case. You know, you can relax the Gaussian noise assumption for PCA. There's a nice paper that does that. You saw this other paper by Yang doing that for the population genetic data. You could do that also for CCA. People have looked at, you know, one of the things about this model, you're assuming that you have the same noise in each direction. You can relax that. You can do regularized CCA. Like a lot of variants of this. As I pointed out, this combination of PCA and CCA, which we did, I think it's going to be very, very powerful for a quick and fast examination of multimodal data. This point really is not mine. It was made originally by Charlotte Sonesson, as I said in her paper. I'm just a messenger. I think it's a really good idea. I think I'm really out of time, so I'll stop there. Thank you very much.
We do have time for a couple questions. Yeah, go ahead.
Could you go back to the PCA plus CCA projection plot, like, and explain how like the steps how it is made? Could you explain again?
How this picture is made?
Yeah.
Yeah, okay, good. So there are two data sets here, first of all. There's an expression matrix and a genotype matrix. And I told you that the points have to correspond. And here they correspond because we have them for the same individuals. So for individual 1, we have the genotypes. For individual 1, we have the expression data. So first of all, that's the input. Now what we've done here is we've done PCA first on each data set independently to higher dimension. And one of the questions is, you know, what dimension should you pick for your PCA? That's always a question. And, you know, we looked at this a little bit, not that much, but yeah, we do a PCA first. Once we've done a PCA, we've reduced the dimension a lot. And that means that now we're going to choose a projection for each one of these data sets. And we have a lot fewer projections because we're in a lot lower dimension. Now we're going to choose a pair of projections first that will maximize the correlation along this axis. Now this axis here is called the first canonical coordinate, if you like. That's one kind of, they go by different names. And once we've done that, then we find an orthogonal direction where we again try to maximize the correlation of a projection of both the data sets in that direction. So that's what we've done here. And you know, that produces this projection of the points. This is the projection from the expression data. You can actually project, there's two different, there's the original expression data and the original genotype data. We've projected the expression data down onto this plane. And so essentially, you can intuitively think we've taken the points in expression space and tried to project them so that they're highly correlated with the points from genotype space. And we know that the points in genotype space are representing the population structure, so we've sort of extracted that projection here. And I haven't shown you more results here, but this is kind of useful because first of all, you can now go and look at which genes constitute the direction here, and you will see genes that, well, we find out that these, they didn't have to a priori, but we find out that these are genes that vary by population. So they're interesting genes in that populations express them differently. The other thing you can do with this projection, and why I think it's going to be useful, is if you're going to now do statistical genetics with expression data, we have a linear projection, or linear map, I should say, that you can sort of remove this component of it and look at the orthogonal piece if you're not interested in a population structure. That's kind of the analog of what people do when they remove population structure in genetic data. So that's all it is. But, you know, there are probably other projections that reveal population structure, not the only one. And we actually explore that a bit in our preprint. But I hope that answered your question. So, yeah.
So this is expression data on the first.
Expression and genotype, yeah. Yeah, it's the expression data projected onto the canonical correlations. Yeah. Yeah.
Another question?
So if one of my matrices is like sparse,
Yes.
and also if I have like features within my two data sets are correlated in some way, so those are two different aspects. Would CCA still be able to capture that, or would we have to use regularized or any other?
Okay, it's a very good question. So a couple of caveats. So first of all, you know, the whole point of CCA is to identify the correlation between two data sets. So that if you don't have that, then there's no point to doing CCA. That's what you would like to have, and that's what you would like to identify. You might want to identify it because you want to see the correlation, or because you want to remove it, which is the application in the single cell RNA-seq. Here we kind of want to look for it, there you want to remove it. As far as, you know, dealing with sparse matrices and so on, well, you know, there's a number of technical caveats here that I've glossed over today. Let me give one example really quickly. So, you know, if you're going to PCA, I had a picture of it here. Yeah, like, you know, when you do PCA, your data lies in some dimension. And you project it to a lower dimension. But it matters what rank your data is in. For example, let's say my data, these black points, are in 20,000 dimensions. This is expression data. But miraculously, they're lying on a plane, on this plane, in 20,000 dimensions. Then I cannot PCA into 3 dimensions. Because if I would try to do that, I'd have many, many answers that are equally good. You know, if you can imagine, if you have 100